Hello everybody, this is Jingying,
Jingxuan,
and Jiayan.
Today we'll be introducing to you a drug named Paclitaxel and how it was discovered and further developed to the drug it is today. So in this video, we will be covering what is Paclitaxel, the administration of Paclitaxel, the timeline of drug discovery and development. We also have a drug discovery video, slides on drug development, the difficulties faced in development, as well as the side effects of Paclitaxel. So, firstly, what exactly is Paclitaxel? Paclitaxel is an anti-cancer chemotherapy drug. Paclitaxel is classified as a plant alkaloid, which refers to any class of naturally occurring organic nitrogen-containing bases derived from plants. A taxane, which is a class of drug that blocks cell growth by stopping mitosis, and an anti-microtubule agent in which paclitaxel binds to tubulin to prevent the polymerization cycle and hence prevents cell division and inhibits growth of tumors. Paclitaxel is also used for the treatment of various cancers, such as breast and ovarian cancer. It has also been used in Kaposi sarcoma, which is cancer that causes patches of abnormal tissue to grow under the skin. Now, we'll be talking about the administration of paclitaxel. Firstly, paclitaxel is given intravenously into the vein since there is no pill form. Next, paclitaxel is typically administered once a week or every 3 weeks. Next, the amount of paclitaxel given depends on many factors, such as the patient's height and weight. Next, paclitaxel is also an irritant and can cause inflammation of the vein to which it is given. If the medication escapes from the vein, it can cause tissue damage. Lastly, this chemotherapy drug may also cause severe allergic reactions and require further medication. Next, we'll be looking at a timeline in which paclitaxel was discovered.
This extraordinary story of discovery begins in 1955 when NCI created the Cancer Chemotherapy National Service Center in response to a call for increased cancer screening and treatment programs. In 1960, NCI expanded the center through a partnership with the U.S. Department of Agriculture, collected bark and other samples in 1962. 2 years later, scientists who were under contract to NCI confirmed the anti-tumor activity of paclitaxel. In 1939, through a grant from NCI, Dr. Susan Ben Hotsmith at the Albert Einstein College of Medicine of Yeshiva University discovered how paclitaxel works. It is found that the compound is an anti-mitotic agent that blocks cancer cell growth by stopping cell division, resulting in cell death. This was an extraordinary discovery as clinicians were struggling to treat patients who were developing drug resistance or not responding to available treatment options. Paclitaxel was proving to be a very promising discovery in cancer treatment. Then after, clinical trials of paclitaxel began in 1983 with a series of Phase 1 trials at institutions around America. The Phase 1 trials demonstrated a series of characteristic toxicities, some of which required considerable effort to overcome. They include several type 1 events with dyspnea, also known as the shortness of breath, bronchospasm, hypotension, and others. The majority occurred on the first or second dose and began almost immediately on initiation of the infusions. These reactions were possibly mediated by the direct release of histamine and other vasoactive substances in a similar manner to radiographic contrast agents. Other toxicities, which have been commonly seen, also include peripheral neuropathy. In 1991, NCI partnered with the pharmaceutical company Bristol Myers Squibb for the commercial production of Taxol using a semi-synthetic form of the compound. A year later, the Food and Drug Administration approved Taxol for the treatment of ovarian cancer and in 1994, the treatment of breast cancer. Next is a video which will further explain the discovery of paclitaxel.
From YouTube. Good afternoon, America. This is Jingxuan. For Science and Healthcare News, there has recently been a new discovery of a potential anti-cancer drug, Paclitaxel. remember the site where the Pacific U tree is. Here, USDA botanist Arthur Barclay is obtaining samples from the bark of the Pacific U tree, also known as the Texas brevifolia. Hi, may I know what you're doing now?
Collecting samples.
What are samples for?
Research of possible cancer cures lah.
And so what are the driving factors of this research?
Increase cancer screening and treatment programs. Not for cancer, for you ah.
Can you give me some examples of the species and plants that you have collected so far?
Hey, I tell you ah, I stay here every day from class already. I dare trigger. You don't piss off anymore. And you, firm some more. Stop from here.
One eternity later.
American chemists Monroe E. Wall and Mansuk Wani, who under the contract to NCI at the Research Triangle Institute in North Carolina, discovered that extracts from the Pacific yew tree were toxic to living cells. Dr. Wall and his colleagues then isolated the most cytotoxic compound from the bark of the tree and called it Paclitaxel. Hi guys, so now we have an exclusive interview with Dr. Wani and Dr. Wall. So let's go. Good afternoon. May I ask what the recent discoveries you have found about Paclitaxel?
2 years ago, in 1977, Paclitaxel has shown efficacy in mouse models, and NCI confirmed Paclitaxel's anti-tumor activity, and the drug was selected as a candidate for clinical development.
Right now, through a grant from NCI, Dr. Susan Bear Horwitz at the Albert Einstein College of Medicine of Yeshiva University discovered how Paclitaxel works. Dr. Horwitz found that the compound is an anti-mitotic agent that blocks cancer cell growth by stopping cell division, resulting in cell death. Even more exciting is that Paclitaxel worked to prevent cell division through a different mechanism compared to other anti-mitotic drugs available at that time.
May I ask what kind of exciting mechanism it is?
So, as you know, during cell division, microtubules depolymerizes to form tubuli, which repolymerizes to form spindles, which pushes cells apart, correct?
Mm-hmm.
So, Paclitaxel is actually special in that it binds to tubulin to prevent polymerization cycle, and hence, this will prevent cell division, and this will inhibit growth of tumors.
This was an extraordinary discovery, as clinicians were struggling to treat patients who were developing drug resistance or not responding to available treatment options. Paclitaxel was proving to be a very promising discovery in cancer treatment.
Hi, thank you for your time.
Thank you.
Thank you for your time.
Thank you. Bye.
So hi, that's all for my interview with Dr. Wall and Dr. Wani. So yeah, thank God they aren't like that botanist from last time, you know. So yeah, that's all from me, and Tisha signing out.
Now, I'll be going into the specifics of drug development. In 1987, NCI-funded researchers observed a dramatic and prolonged response to Paclitaxel in a patient with cisplatin-resistant ovarian cancer during a phase 1 clinical trial. These initial results gave doctors hope that they might have a new tool to treat ovarian cancer other than traditional approaches such as radiotherapy, which is not as effective. In 1989, 10 years after, an impressive 30% of patients with advanced ovarian cancer responded positively to Paclitaxel treatment. In 1991, NCI partnered with the pharmaceutical company Bristol Myers Squibb for the commercial production of Taxol using a semi-synthetic form of the compound. A year later, the Food and Drug Administration approved Taxol for the treatment of ovarian cancer, and in 1994 for the treatment of breast cancer. Today, Taxol remains one of the best plant-based cancer treatments available. However, throughout the various stages of discovery and development, there were some difficulties experienced. Firstly, the drug has no activity when given orally and is poorly soluble in water. A number of approaches were evaluated before settling on the chromophore-EL, which solubilizes the compound and allowed dilution of the nearly saturated solution. This eventually proved ideal for clinical use from a pharmaceutical perspective. Another difficulty was the high incidence of acute hypersensitivity reaction caused by the drug. Studies were then completed using prolonged 24-hour infusions and a pre-medication regimen similar to that used for contrast agents. Subsequent clinical trials were also delayed because of how slowly Pacific yew trees grow. Moreover, large amount of bulk is needed to produce therapeutic doses of the active ingredient paclitaxel. The slow-growing nature of the Pacific yew and increasing demand for the compound led to another roadblock. The drug was very expensive to manufacture and the ecological cost was too high, as harvesting the bark kills the tree. There were also complexities involved in developing a synthetic version of the compound, but they eventually took another approach and developed a semi-synthetic version of the compound instead. As you can see in overall, it took a full 39 years for the discovery and development of the drug.
Lastly, we'll be touching on the side effects of paclitaxel. Firstly, paclitaxel may cause low blood count, hair loss, arthralgias and myalgias, which is pain in the joints and muscles, peripheral neuropathy, which is numbness and tingling of the hands and feet, nausea and vomiting, which is usually mild, diarrhea, mouth sores, and hypersensitivity reactions such as fever, facial flushing, chills, shortness of breath, or hives. We have come to the end of our video.